Welcome to the biology class once again, students. So in the last class we learned about the structure of protein. We understood the various levels of the structure of a protein. We started with the primary structure and we learned that primary structure is the most simplest form which is possible, which is present in all the proteins. So primary structure is nothing but the arrangement, sequential arrangement of amino acids, and this arrangement is governed by the genetic material. So DNA will cause the formation of the mRNA, messenger RNA, and messenger RNA will carry the message. That's why it's called messenger RNA. It will carry the message of which amino acid will come first, which will come second, third, fourth, and this number of the amino acids required and their sequence is already fixed by the genetic material. And two amino acids will be joined with the help of a bond which is called as peptide bond. Please mention in all, I have found in assignments you all mentioning covalent, it's correct, but the real name of the bond is amide bond or otherwise peptide bond. Now this whole primary structure is made up of a number of amino acids in a fixed position in the fixed number also, and the long chain will have two terminal. First terminal is the left terminal, which is represented as the N terminal because the amino group of the first amino acid is going to be free on this side. That's why it is called as the N terminal. And the last amino acid will have its carboxyl group free. That's why it is called as the C terminal. So there are two ends of the primary structure. Left terminal as N terminal, right terminal as the C terminal. First and the last. Remember the bond formed is amide bond or peptide bond, which is CO and NH, which we have learned in the first class itself. And the distance between two peptide bonds would be 0.35 nanometer. Now primary structure is, because primary structure is going to be a very long one, reason there are some amino acids made up of, there are some proteins made up of only 31, 21, 40, 45, this number of amino acids, but there are some which are made up of hundreds of amino acids. In that case, this primary structure needs more stability, plus it will undergo further coiling, or you can say two multi-dimensional structural changes will come. Now those changes will develop the formation of the secondary structure. So what is secondary structure? So primary structure is common to all, it is present in all proteins. Secondary structure now. So the next level of the structure is the secondary structure. Now secondary structure is stabilized with the help of hydrogen bonds. Now those hydrogen bonds can be intra-polypeptide or they can be interpolypeptide. Then we learned that the secondary structure is of three various forms: alpha helical form, beta pleated form, and collagen helix form. Now alpha helical form is called alpha because it was first to be discovered. Later was the second one was the pleated form, that's why it is called beta pleated. And very recently an Indian scientist discovered the collagen helix. So the alpha helical structure is common to those which are able to undergo coiling, and this coiling will ensure that every turn will have 3.6 amino acids, or you can say roughly 4 amino acids per turn, and every first amino acid will develop hydrogen bond with the fourth amino acid. So it's gonna be like every amino acid is going to participate with the fourth one, then the fourth one. So all amino acids are involved with their amino group and their carbonyl group. There will be formation of hydrogen bond. Amino group and the carbonyl group, there will be formation of the hydrogen bond, and this bond stabilizes the structure of protein to the next level, that is the secondary structure. Now this bond formation occurs in the case of monomeric protein. This bond formation occurs in the same chain, that's why it is called as intra-polypeptide hydrogen bond. But in the case of the oligomeric or multimeric type of proteins, it occurs in the interpolypeptide hydrogen bond because different chains will develop hydrogen bond. We learned that these chains can go, when there are more number of chains, we're talking about beta pleated now. So if there are more number of chain, they may run parallel or they may run anti-parallel. Parallel means the N terminals facing each other, anti-parallel N terminal facing the C terminal of the other chain. We learned about the helix breaker also. there is a nice symmetrical systematic manner of the helical form. But sometimes what happens, if a primary structure carries an amino acid which is called glycine or proline or hydroxyproline, then these amino acids do not develop the hydrogen bonds. So they are called as the helix breaker, because now the secondary structure would become a random coil. So coiling is very symmetrical in these, but the coiling will start breaking, and this breakage is because of these glycine, proline and hydroxyproline amino acids. Now because lately it was discovered that collagen carries a greater percentage of glycine, proline and hydroxyproline. That's why another type of structure is found in glycine which is not alpha helical or beta braided, so it is called as the collagen helix, where 3 chains coil around in such a manner that it develops a different type of structure, so it is called as the secondary structure. Now the next thing is the tertiary structure. The same, okay, we learned in the last class that fibrous proteins will have primary structure and secondary structure. They will not have tertiary structure or a very little degree of tertiary structure. Now those which are globular proteins or those which are multimeric or oligomeric protein, they will have primary structure also, secondary structure also and tertiary structure also. So tertiary structure happens because of further folding of the secondary structure, and this develops a large number of other bonds also, like hydrophobic bonds I told you in the last class, disulfide bonds we have already learned, hydrogen bonds, ionic bonds, van der Waals forces. So many forces will participate in stabilizing the tertiary structure. And the last structure we learned in the last class was the quaternary structure. Now what is quaternary structure? If a protein is monomeric, it will have only primary, secondary, or it may even have the tertiary structure. But if a protein is multimeric or oligomeric, it will have primary, secondary, tertiary, as well as another degree of the structure which is called as quaternary structure. So for example, hemoglobin has 2 alpha chains, 2 beta chains. So these chains, alpha chains has 141 amino acids. So each chain will have its primary structure, secondary structure and tertiary structure. Then here also beta chains will have 146 amino acids. Each chain will have its own primary structure, secondary structure, tertiary structure. And finally these proteins, 4 proteins, because 2 alpha and 2 beta, these proteins, these chains also called as protomeres, these chains will interact now to finally give the quaternary structure.